This presentation covers Nuremberg and Mathai and Nuremberg and Leader's experiments on the genetic code. What people knew before their experiments were that ribosomes could make proteins from mRNA and were formed by amino acids, but they didn't know how the genetic code worked. So Nuremberg and Mathai discovered 3 of the 64 codons. Those were the poly U, poly A, and poly C. How they set up their experiment was the test tube that they used had all of the components of a cell, but no confinement. They added radioactive amino acids. To this, they were able to compose their own RNA sequences and add it to the unconfined cell. They had to repeat this experiment 20 times because that's how many amino acids there are, and find which amino acid was created by adding in their poly U sequence, and this ended up being phenylalanine. The only time a sequence formed a protein was when phenylalanine was the amino acid associated with the poly U chain. They next tried to find out how many bases they needed to make each amino acid. The math showed that they needed at least 3 bases in order to make the 20 amino acids, because if there were only 2 from the 4 nucleotides present, they were only able to make 16 different combinations, but with 3, they're able to make 64. In addition to this, the 2 nucleotides, when experimented on, showed no activity. However, 3 or more did. They also found that tRNA requires an RNA in order to stick to the ribosomes. As for Nuremberg and Leader's experiment, it was very similar, and they went on to discover the rest of the 64 codon combinations. What they did was they added the short RNA sequences to a mixture of tRNA and ribosomes attached to amino acids. They used the different radioactive amino acids again to tell if that particular amino acid would bind to the ribosome. If the tRNA, ribosome, and codon were all bound together, then they would be caught by a filter paper, which is shown in the diagram here. However, if they are not bound together, then the tRNA and RNA are able to pass through paper when washed. So whenever the RNA is able to be bound to the ribosome and tRNA to form a new amino acid, that's how they were able to discover which RNA sequence coded for which amino acid.